Chris today to talk to us about work he's doing with integrating microscope work and artificial intelligence, AI on the edge. Very similar to work we're doing here at Fermilab. He was trained as an analytical chemist, is that right?
Yep.
But was always the go-to guy for making things work in the lab. And we all like people like that. And he's wound up forming a company in collaboration with his brother in Michigan. And he's going to tell us about what's going on.
Yeah.
So let's welcome Chris.
Well, thank you, everyone. Thank you for the introductions. Yep. My name is Chris Keel, and I'm going to be talking a little bit about scaling quantitative imaging platforms using machine learning for mostly electron microscopes, but pretty much anything that takes an image. As mentioned, my background is actually in analytical chemistry and computer science. Basically, I've been writing scientific software and getting instruments to work for the last 20 plus years. Then the pandemic hit, and I was talking with my brother, Kevin. I am the older one, he is the younger one. But he is a professor at the University of Michigan in the nuclear materials characterization and microscopy department there. And he is the one who's been drawing boxes around black dots. So we're gonna see a lot of images of black dots today. I apologize for that. There will be some more interesting images, I promise, but you'll see a lot of black dots. But he never wanted to draw boxes around those things ever again. He's probably been doing that for a decade plus. And has done at least 10,000 images that he's tried to do by hand for that. So we were talking, and I wrote some software. I showed him a demo, and here we are now into the future, and we've formed a company, Theia Scientific, on that. But some of the stuff you'll see today is some of his research and some of his work that he's done from the University of Michigan. But we'll go with that. So to really figure out where we're going and what we've done, I need to kind of start with Kevin's research and kind of give the world's worst primer to radiation effects. This is normally the part where Kevin will give the talk. He has a whole class, a semester long, on all of this. So I will try and do this in about 30 seconds, so bear with me here. So we've got radiation effects in materials. What Kevin's research and what Kevin has been doing is they're trying to figure out what kind of materials should we build the next generation nuclear reactors out of. And this includes also the fusion systems and the fusion energy that's been a big thing recently, part of the Department of Energy as well. But also you have your light water reactors, you have your Gen 2s, your Gen 3s, you have your molten salts. All of those things have materials, and they all have to have some kind of containment on there. And they are, of course, an extreme environment. We talked with some people today as well that also deal with extreme environments. This is extreme because you have radiation, you have pressure, you have various stresses, water, corrosion, all that wonderful stuff sitting there in your reactor. And they want to know, well, what should our materials be when it comes to that? And we're kind of in this space right here when we're looking at the different things. There's a whole field on fractures, corrosion. As Kevin says, corrosion is a dark art and he doesn't want to deal with it. So we won't talk anything about corrosion. We're mostly in this radiation-induced segmentation, radiation creep, and what does radiation do to your materials as you're building new reactors. So with that, we will zoom all the way down on our scale down to the level that many of you work at, which is in individual particles or atoms, or at that level of scale. And then throughout this presentation and throughout our work, we'll scale all the way up to much larger systems when it comes to that. So we start off with, we have a particle. Some of this may be a review for some of you, some of this might be new, but we want to make sure that we're all in the same process. You have a lattice of your material, you have your various atoms, you have this particle coming into it, and it's going to hit, right? And that's going to cause some collisions inside your lattice, inside your material, and as that moves through the system, you start creating more and more defects or voids or cavities, and you want to then quantify all of this. You want to quantify what's coming off of it, you want to quantify the spaces that are coming in between all of that, and that's going to tell you whether you have a good material or a bad material, either for your reactor vessel or for your fuel that's going inside the reactor, and so on and so forth. For that. But in addition to characterizing that and having these induced effects, we also want to simulate this, right? Because we can't simulate, well, we can simulate, but we can't have a molten salt reactor for 100 years and then see how it works to build the next one, right? We have to kind of get it right the first time situation. But this is a simulation of an atom of what we just saw through those slides, and you can kind of see this is what you're trying to capture, right? And they're trying to capture this, though, of course, experimentally, and we're trying to mesh those two together, the theory, the simulations with the experimental, the empirical evidence to, again, decide what is going on. And it turns out that in this research and in this work that the microscopy and image analysis works pretty well, because we're starting down here with your primary damage and it's scaling as well. And what we're trying to look for is the growth of those voids. Do they come together? Do they split apart? How does that work? And then you start adding in the different alloys and the different material compositions, and you start getting this matrix of information to decide, again, is it good material or bad material? And what we're ultimately looking for in our information is sort of the size, your number densities, and your shapes of those damages or those defects in your materials. So how do we characterize this? How do we look at radiation damage from an empirical standpoint? And we have, again, here's our scale. So we're talking about scale today in a variety of different ways. And what we're mostly started with and where we kind of looked at was TEM, or transmission electron microscopy. That's mostly because that's what Kevin's research has been in, and that's what Kevin did his thesis on. And this is Kevin circa 2009, 2010 on a Joel TEM at the University of Wisconsin. So this is where we're kind of focusing, and this is where our, if we're going to have a little bit of business talk today, this is our beachhead market, right? So this is where we're entering into the market from, and this is where we're going to get all of our pictures of black dots. So that's how we're going to characterize the material, but we also need to induce those damage. We need to create those defects in a controlled way that isn't involved sending somebody into a reactor, holding the thing up to the fuel and saying, okay, what happened? So we need some other way to do that. And what Kevin is the director of at the University of Michigan is the Michigan Ion Beam Laboratory. This is one of 10 facilities in the world. I think it might be the only facility that has 3 beam lines on it. If he was here, he could correct me. But it's also the only TEM microscope that has 3 beam lines like this. So they started off with this one here, then they had added another one, and then they added the Wolverine, which is in the background. The big giant one, that one was actually with a collaboration with TerraPower. But they basically can direct these beams, and they can direct all of them down to the transmission electron microscope down here at the end. So the idea is that you're going to place your sample into the transmission electron microscope, we're going to open the door to one of these beams, and we're going to induce damage, and we're going to watch what happens, just like we saw with that simulation over time. This is a unique facility because they basically can create ions and particles from anything on the periodic table. They have, I think it's 5 full-time staff members that handle it. This is a modern in situ TEM system, modern because it's actually from 1974, but this is pretty much, as Kevin likes to say, the physics of electrons haven't changed, so this is still what you would do if you're going to build an in situ TEM. This is what's on the end of it. The microscopes have gotten better. You're no longer developing film, you have digital images that you're taking. This one is actually at IVEM, which is we'll call it a sister facility over at Argonne National Laboratory. We've hooked up to this system, but you can see you have a variety of different decades of technology being coupled together for these instruments and these systems. But the beam is coming in here, and then you have your electron beam, your sample is sitting down here, you have a bunch of magnets and everything to direct it, very similar to some of the facilities here as well, but you're then taking the pictures and looking at that. So this is a picture and a cutaway of the IVEM in situ TEM. And then this is the one at Michael, which is, again, up at University of Michigan. So here again, you have the beam coming in from over here at a certain angle. The angle is important. You have your column here. You have your graduate student or postdoc. This is Priyam. He was with us at the beginning of all this project. This is a little preview of our software running on there. This was actually the first time we ever did an in situ TEM ion radiation with ML-assisted microscopy image analysis. So,
is that the image in the lower left?
Yes. Yep. You can tell because it has the black dots.
And you
can
got a
see a lot.
question?
Yes.
So, having been in a radiology theater before, usually when you're around anything like this, you wear lead and all this other kind of stuff. What's your fluence that you don't have to worry about any radiation to an operator?
Yeah, so it's all enclosed, it's all in the shielded all the way through the whole system, and then their DPAs are low enough that that's not an issue. There is an end station back in one part of it. Up here, you can see they've got the shielding and stuff from the wall over here, right? So this is a special one where if you're doing higher DPAs, you would run over there. That's not what, but they don't do that over with the in situ TEM. And then they also consider the in situ TEM then sort of contaminated afterwards, but it's never, the samples are 100 millimeters by 100 millimeters, like, or 10 millimeters by 10 millimeters, I forget what it's down to. Samples are small enough.
What does in situ mean in this case?
In situ means that the beams are coming in as you're running your electron microscope. So ex situ would be the older style where you would be at one of these beam stations, you would expose your sample, you would take it out of that station, and then you would put it into your microscope after the fact. The in situ TEM is special, and the in situ part is that you are exposing it and doing the damage as you're watching it happen. So where were we at? So I promised black dots, here are some black dots, right? So this is an example of an experiment that was done. You can imagine you're sitting at that microscope, your pream sitting there watching it happen, and we're looking at these black dots form and change over time. And you would then sit there, and an experiment like this would take anywhere from 4 to 8 hours. It could also go, you could do an exposure on the beam and then irradiation damage up to 2 weeks even. So a little bit longer time scales when it comes to this, but you can now imagine that there's maybe at least 2,000 frames here, and you have to identify all those black dots in that. No one wants to do that, so what do we do next? Yes?
What was the frame rate?
So that one was actually a time-lapse video where they took an image basically once a minute, and then they compressed it all down to 2 minutes.
I'm just trying to square away that with your video where the physical processes were happening in the picosecond time.
Yes, so one of the things though is that that was the simulation part, and they're trying to figure out that they can't at the moment because your detectors are going at, well, they have new detectors that are coming out that will be up to 500 frames a second to 1,000 to 10,000 frames a second. So they will be able to capture things like that, but their current workflow, which we'll get into in just a moment, doesn't allow them to capture that level of event. So you can only look at what happened after the fact sort of situation. And what we're doing with the beam and what we're doing with the in situ TEM, remember, is we're trying to simulate basically 60 years of a material running in a reactor, right? We're trying to, so the individual, we'll call it sub-microsecond phenomena is not something that this current level of scale they're interested in. They're mostly at the materials level. They're looking at, will this last long enough for us to run the reactor for 60 years? It's kind of a difference. Here's another example of an in situ TEM irradiation. Again, we're at the Mybel facility. This time, that's our software up there, or that's our technology running there. But this is the control software that they have. This is their the microscope control software. This is the camera detection software and control for the camera. This is their beam control, and then this is an extra one because they need to have 5 monitors because I'm sure they wrote a grant for that. Had to show it off, right? But you can see, you can imagine you're sitting here and as an untrained person in this sort of thing, I have no idea what I'm looking at. There's just a bunch of black dots. We have to somehow convert that into data. Yes. Yep.
If you go back to the video,
Yeah.
do you have a consistent contrast ratio here? Because, for instance, there are actually shapes that are coming in and out of blackness.
Right. So one of the things that they're doing is they're constantly playing with the focus, essentially, is where that's coming in, and they play with the contrast on the microscope as they're running it. Because the microscopists are basically trained to make the best image possible all the time, to the sacrificing of everything else. And so any sort of variance you have in here that you're seeing, the big question ultimately is that real or not, or is that an artifact of the microscope? What's going on, that stuff. So there's also, you can see it kind of jitters. So there's drift correction because as the radiation hits it, as the beam hits it, you're heating it up, it expands, it moves the sample, it causes all kinds of problems on that. So they spent basically the better part of 4 years just trying to get all of the parts to move together so that they could take just even these images and keep it kind of constant. So one of the things that we're working on that they want to compensate for is to track all of that and actually add in drift correction through our system for it. But, so one of the challenges that has been, and we'll get into this in just a moment, is this varying contrast causes problems then obviously on the back end for the image analysis, right? Because like, how is thresholding going to get you through that? Because your contrast is always changing, your background is always different, right? And so you can't do just a straight up background subtraction.
So not to bring up radiology again, but this is a solved thing in radiology.
Yes.
But anyway,
What's the solution there?
the solution is you do slices, right? And you basically then you tune to a very specific density of some fluid in your body, and then you apply that as a consistent correction and or calibration.
Yeah, so they are doing part of that in the past. It is part of the processing on this particular system here.
Okay.
But one of the questions they always have is this a good sample? Right? So, because the other question is, if it's a 4-8 hour, week-long exposure,
Sure.
do we have to go that long? And how do we know? And if we can't know until the very end, that's going to be a problem, right? So that's one of the issues. But there is some image correction and image processing that is done by the detectors and done by this software here, but they have not integrated any ML into it, and they don't tell you, is it a good sample or bad sample?
Okay.
All right, so what they do, because they don't know, is they will just collect thousands and thousands and thousands of frames, and then they sit there and they will have 1 cup of coffee and they get maybe a couple of frames done, and then they get bored and they leave, or they have an email, or they call, or they get tired, they fall asleep, whatever. So they end up only doing a couple of them over time, where they think that there's good things. So there might be missing those events that are occurring in between the frames if you're going faster. And here's an example that they did. They published a paper, they threw away 99% of their data just because they couldn't process all of it in the amount of time they wanted to do it in, and it took them 2 years to publish that. So what this ended up doing, and this kind of came to the crux of why Kevin and I started the company, was we should be able to do that faster, right? We should be able to go we should be able to analyze all of these frames, and we should also be able to not take 2 years to get to the data that we want, right? So this is the overall workflow then for this type of research and this type of work. You start off with your acquisition on the microscope. We're going to count all the black dots. We're going to get the size, the morphology information from that. We'll call that quantitation. We're going to convert that into some kind of plot. We'll see where our maximum or minimum is, or we'll make our analysis, and that will give us our discovery, which gives us our Nobel Prize, right? This is the general sort of workflow that they're trying to work on. If this is the higher level version of it, we can zoom in then and look at what is it just from each step of there, what is going on. And so this is our current workflow for working on a transmission electron microscope. You start off with your acquisition. You will collect your images. You will collect thousands of them because you're not sure if that was a good image, a good sample or not, and you don't want to miss anything. You export that. If you're unlucky, you will burn that to a DVD. If you are really unlucky, you will put that onto a floppy disk and take that across the lab to your office. You will generally import that then into some other workstation that has more horsepower, compute power essentially, than the microscope controlled computer, because all these microscopes were bought in the early 00s and they were dipped in concrete and you were not allowed to touch them, and they're all running Windows XP. Right? So you have to use some other system, and that maybe is the one computer that has the dongle for the software that you need, that you paid the license for back in 2005 on, and you'll import your data, and that's the one that has the decoder that you need for the proprietary data to import onto that. You'll run your image processing, you'll do your feature detection, quantitation, you'll then export it to your laptop, and as you're going to the conference, you will put this all together. That's where you'll do your aggregation, and you'll finally do your interpretation and discovery and present it to an audience, correct? So if we look at this, what all the microscopists and scientists want is this part here, right? But there's all of this relative time that's going in there, and there's this big part here, right? You've got all that data and you're trying to do the analysis. So if we can shorten that time, we might be able to do that. And if we look at this thing, this workflow is obviously very laborious, it's inefficient, and there's inherently bias towards the microscopist's expertise. But there's also another key part in this current workflow, and you can see that with the red lines, right? First of all, the export-import. Okay, I put it onto my thumb drive, I walk back to the office, oh, there's an email, oh, I got a phone call, 6 months later, I finally import it into that workstation, right? And there's 2 jumps for that. So you could talk about, hey, now we're trying to get to that 2 years that occurred, right? But there's also, along the way, you have this negative feedback loop. And we have one occasion where we're talking with one lab and one person, 2 years afterwards, they're down in this phase here, and they found out they collected the entirely wrong data. They were looking at the wrong area while they were on the microscope. And so they had to go back, and it took them then another 2 years to go through this whole process all over again. Right? Now there's that last part in this, which is inherently biased towards a microscopist's expertise. So, in that case, if it did it lock up on it, it will not move to the next slide. There we go. Right, so they set out and said, okay, let's look at that bias, because there's bias in the models out there, there's bias in, and there's been this long-standing sort of mentality that you can never do better than a microscopist, and that the trained expert is always going to be better than anything else that you can do. And so they did a round-robin experiment. They sent out a variety of images, the same images, to a bunch of different researchers and asked them to quantitate the images by hand, right? Let's work that workflow, let's go through that process. And we can see that there's actually some bias, and there is some, we think that this person ran out of coffee or something, or there's also some inherent variability in the system, because you get two microscopists in the same room, they're never going to agree about what's a black dot or not. Right? It's just everyone's going to have their own opinion about what it is. They're also, maybe this user changed the contrast afterwards, right? They applied some other image processing algorithm to it that they just always use, and they've never defined it, or they never put into their papers that they did it that way, but that's what they always do. So it allows them to see things in a little bit differently, and they count more or they count less, depending on that. So there's a lot of variability and bias then in that workflow from that system. So you're an enterprising graduate student or intern, and you've been tasked with analyzing 10,000 frames to get to that one plot so you can publish your paper, and you're like, there's no way there's not enough coffee in the world to make that happen. So you eventually set out to do some computer vision, right? How can I automate this, right? And the question ultimately then is, you start off and you say, well, what is out there? And you start off with the thresholding, you use image J, you start playing around with that stuff to kind of see what it is, and you ultimately spend a whole summer or more trying to get your one model to work on your one workflow to work on one particular image, and it works really well for that one. Then you apply the second one, does not work at all, right? And so now you abandon that, and you eventually go back to manually annotating all those images, right? And because now we're sitting here and you're looking, I apologize, these are not black dots, you want the white dots in this one. That's maybe 200 to 300 of them in there that you're trying to grab, right? So there's been attempts at this over the years, and we all know now, and this is a little bit different because 4 years ago this was not the case, but now we know that there's all of these other methods, and the deep learning, machine learning has sort of, I don't want to use too many hype words, but has revolutionized what we're looking at now, right? Because it can account for the contrast background changes. It can be better than those traditional methods of computer vision, or it can at least be used to augment that, and you can have a hybrid one, and now you can get it to work on all of your images, or at least get to the 99% of your images are being quantitated as opposed to being thrown out. So I'm sure many of you are familiar with this, but around this time, right, we get the LXNet, they really kind of kicked all this sort of stuff off. This is actually a little old now because it only goes up to 2019, but you have your various deep learning models, and the one that we're going to look at the most and use the most is probably YOLO, but RCNN was a big one, the fast RCNN, faster RCNN, mask RCNN, on and on it goes. You have your one stage and your two stage. So now we're getting to that point where, hey, maybe we can actually start doing this in an automated way, and we can start applying this to electron microscopy, and I don't have to draw boxes around white dots or black dots anymore. And it turns out that yes, you can start doing this in that thing, which leads to a very quick side note and side story. All of this for me started because of a conversation that my brother and I had, which is, I saw his images and I was like, hey, your black dots look a lot like my black dots. Maybe there's something we can do there. I have a solution for my black dots, can it work with yours? And the answer was yes, because that's why I'm here, right? So if your black dots look like my black dots, reach out to me and we'll see if something can happen for that, right? But what we're getting now is we're getting close enough that our models can start doing all this quantitation for us. We can start shrinking that time in that current workflow and putting that together. So the current workflow kind of looks like this for machine learning. This was the YOLO v5 version 6 of a v5. Yes, I know naming is a problem. But this one was actually with some work with the University of Wisconsin. And I believe that image is actually from, yep, it's from IVM, so that's from Argonne National Laboratory that's also been working with us to put this together. So I'm going through this one a little quickly, just because I'm pretty sure everyone kind of knows there's the training stage, there's now the deployment stage, and so on and so forth from that. But this is just showing that, okay, we can do this with in situ data. Now this wasn't, I'm gonna say live, this was a video after it of in situ data that they created and then applied it to it. So it wasn't at the microscope, it wasn't quote unquote real time. I know everybody has a different definition of real time, so, but real time for the microscopist.
But was the speed it was being done the same speed that the data was coming in?
It's about 30 frames a second.
He's asking about the interpretation.
So the interpretation, so you ran this on a video,
Yes,
but the number of seconds it took to do the analysis,
yes.
is that the number of seconds the data was collected for?
No, the data was, again, it was like a time-lapse video of, because it was an in situ TEM. I think this one was probably once a minute as well.
Ah, okay.
And then over the, I don't know the exact specifics on there. It is in this paper though. All right. So one of the things about the speed is that they go at once per minute because because they're like, we can't go, like, we're not going to look at those images if we take any more of that. Right? So one of the things that we've been able to do is enable them to collect faster on all those acquisitions. So real brief recap then of that workflow. So this is the sort of current workflow for machine learning, right? I don't know what's going on there, but whatever. Is so you have your development where you're taking your images, you're labeling it, you're creating training, you're testing, you're evaluating, you then publish it, you publish your paper, and then you don't worry about whoever's doing anything else. That's this part here, which is deployment. So people are creating new models all the time. I'm sure 3 of them were created and published in the 30 minutes I've been talking so far. So the question though is that, what about deployment? How do I get it so that a microscopist can use this model that someone else has created? Right? Because microscopists don't want to create the models, they just want to use the models, annotate their data for them, and move on and get their answers. So really what we're looking to do is we just take a briefly looking at this current workflow. We want to merge this workflow with the machine learning workflow so that we can shrink this time and ultimately shrink some of these larger boxes. And then what we're going to do is we're going to transform that into this workflow. We're going to collapse it into a linear one. We're going to say, okay, can we do it faster? Can we shrink this time and that time and save 80% on that? But can we also turn those red lines into green lines? Can we say, hey, this is a bad sample, let's stop. Let's load up another sample, because we're also paying for time on the microscope in this case, so we can save time and labor on that and cost. But then also, we said, hey, can we do this at the microscope, right? As opposed to doing it in the post-acquisition sort of workflow where we collect it, then we come back to our computers later on and do the analysis. I want my paper, I want my publish my paper button right in my microscope, right? That's kind of what we set out to do in that case. But there are, of course, some challenges to machine learning deployments, right? I talked to some of you earlier today about some of these and commiserating on some of these, and of course you have a variety of standards, and we're gonna maybe add some more standards into all this, but you start off with your dependencies, you start off with the environment that we're in, and then there's the variety of formats that you have to do for the machine learning, but also for the microscopes and all of theirs, and then you have source code and data set management, which includes also not everyone is a software developer and knows what Git is and how to use that in an appropriate way. So you have all of this that you kind of need to do. None of this any microscopist ever wants to deal with, right? So we need some way to deploy these models in a way that doesn't require any code, but also manages all these dependencies in the system and handle all of these challenges. And another part of the challenge, for us at least, has been this, that most of the machine learning stuff that we see from microscopy starts off with a Jupyter notebook, specifically Google Colab notebook, because hey, I Googled it and wow, Google comes out with a Google product to handle the machine learning for me, so I put that in there, I run that. But the question we then always ask is, well, now how do I run it? How does somebody else in your group run it? How does somebody else, how do you transfer that? How do you deploy that model or that training to another system? And it always depends on kind of what level the person is on there. More often than not, only that one person is going to be able to use it. But that was also true for the traditional computer vision systems too, right? There's all these little variables, there's all these different things to tweak and try, and only one person kind of knew how to do it, and you have to give that script to somebody else, and then they're like, well, now how do I use this, right? And then there's also the problem of, well, you have this interoperability, right? We're trying to move from developed to deployed and have people think about deploying this model and giving it to somebody else and giving this code to other people. And then you also have the wonderful, which I could spend the rest of the talk griping about, Python dependency management that you have to deal with, and all of that. And a large part of this has been solved with Docker, but again, no microscopist is ever going to know what Docker is or how to use it. So we need some way to take all of this sort of stuff, package it in a way that other people can use, and we can put it onto a microscope. Right? Because in addition to all of these problems with machine learning deployment, you also have the environment that these microscopes are in. Right? Machine learning sort of really took off because you have the cloud-based stuff. You have Google Colab is in the cloud. You're at your computer. You have internet access. You do not have that. You don't necessarily want your microscope connected to the internet. Again, remember, they're running Windows XP, so of course you don't want them connected to the internet. But you're also not allowed to modify your microscopes. You'll void all your warranties. You'll void all your service contracts. That computer probably doesn't have an AGP port on it that's of quality to put a GPU in there. So you need some other way to bring these cloud resources to the microscope as opposed to trying to bring the microscope to the cloud. And that will then allow you to run the models and allow you to do the feature detection and avoid having to deal with the sneaker net with your thumb drive, right? So with that, we created then the Theiascope platform, or what we call Theiascope. Brief aside, Theia is the Greek tightness of vision, so it's a vision scope. Right? That's where that all comes from. But what we're going to do is we're going to have essentially a Raspberry Pi with a GPU on it, so we're going to have an edge device, and we're going to load all of our models onto that. We're going to have a time series database. If you think about what a video is, it really is a self-contained time series database. Right? Each frame has a timestamp on it, and it's compressed together. It's basically a giant zip file. So let's do the same thing with our database. We'll use a relational database with a time series component on it so that we can relate our microscope control software, and we can relate all the metadata to the different frames and then to the time as things are collected over time. We will connect everything with a LAN port because all the microscopes now do have LAN ports on them or USB ports. We can use the dongle. And we can also provide over-the-air access if you are in one of those special institutions that does allow you to connect to the internet from your microscope. We're finding out that that's happening more and more, but it's through a separate network and it's all that other stuff. But we can then also have on the acquisition PC, all you need is a web browser. So you don't even need to install software on the system because even Windows XP has a web browser built into it. So we don't have to void your warranty in order for this to happen. So we try to make it as low touch as possible, and that you can also then, because we're using an edge computing device, you can carry that with you. It becomes your thumb drive. It walks with you from instrument to instrument. It walks with you from your sample to sample, and you can take it back to your office and you have your data and your models all in one place. So you can do the analysis at the microscope. We get around the problem of the environment, but you can also do it and still maintain your post-acquisition workflow.
Excuse me.
Yes?
So, since you have deployed your model on the edge device,
Mhm.
so like how do you handle the, because your model can be of huge size, so how, do you employ pruning techniques or quantization techniques, something which lightens the model parameters, number of parameters and size?
We don't do that on the device. That is up to
No, before
the user.
itself.
Yeah, you can do that, TensorRT. You can run that through the system. We can actually show you that in just a moment. But that's up to the user. So the idea was that there are groups at pretty much every institution that we talk to that are building models. And what generally happens is the microscopists, the scientists go to them and are like, hey, can you build me a model that will automate this workflow so I don't have to draw boxes, right? And they will develop a model, and then they'll be like, here's your model. And the microscope users will be like, well, how do I use the model? And so then they develop some script or something. Well, you type this code here, use this notebook, and then the microscope users are like, well, I don't want to write code, right? And so then they're like, and then the model developer's like, well, we don't want to support or create a whole GUI or anything. We want to just run. We're more focused on that. So they would do the pruning, and they would do that from that standpoint. And then what we would be is the target platform for deploying the model. And they can just be like, oh, well, we'll just prune it if we want it to fit on there. But we've actually got it so they can work on the edge computing device or just a standard data center grade GPU if you wanted to. So, because one of the things that we've learned is that every microscope has a rack, so you can put your own rack server in there and have, we've done 11 GPUs at the microscope, so, which we'll get into. Right. So, rather than show more videos about that, we can actually escape out of this, and I know demos are always a concern, and we'll see if it works. But, so this is the system. This is this device right here, and at the end of the presentation, you can go ahead and connect your laptops to it, and you can upload your own images, and we can see if it works or not. All right, but we have our interface here. You start off with, again, I said the time series database, so we'll start off with a calendar. All the professors really love this because they could tell if the students were actually doing work or not, because they can log in, they can connect into it, and they can look at it, which is what we will do. We'll do that first. So we can go ahead and we can go back in time, and we can see I've been doing a lot of different experiments on this particular device. It tells me how many images I've collected, where they've gone, so on and so forth. But let's go ahead and take a look at one particular image here. And that's because this is not black dots. This is something else we've been working with Columbia University on, and that's looking at the grain boundaries. And I'm showing this one as an example in the system because they had 30 years of data. They were actually films that they had developed in the late 90s, and they would print them out on transparencies on a projector and draw the boxes around that way, and then scan that back in and use that to do their analysis with. And they had 30 years of that data that they've been doing. And then they finally came along and said, well, no one wants to do that. And so they trained a model, they did it, it seemed to work, and we're like, well, hey, maybe we could run your model on our system and see what happens. And the answer is yes. It does work, and we can do the segmentation. So just picture yourself, you have to sit there and draw this really weird line and fuzzy line around 30 years worth of data before they could publish their paper on this system. And the answer is though that it does work, and we can do the segmentation for it. We can also do the binary image for it, so we can look at it from that standpoint, and then we can also convert that. Right, so now they're getting their grains, because what they really wanted was the grains in between. And they're able to see that kind of system. And what they really truly want are actually the intersections of those points and then monitoring those as they move over time, which is, we're working on adding that in there for them on this process. But they were already fully ecstatic about being able to do that. But this was a pretty good demonstration of the various ways we could also do just the bounding boxes if you wanted, so on and so forth. I don't want to do a whole demonstration of all the stuff that's going in here, but I just wanted to show that particular one because you're also getting your size distribution, you're getting your annotations, we can zoom in on the timeline. For those of you old enough, this is like a VCR. We can move forward in time, we can go forward, backwards in time and see all the different images as they're moving. We can also zoom in and we can look at particular items in our system as we move through the process, and we can go ahead and, like a video, we can play it back. Just like a VCR, scrolling back and forth through that 10,000 images if that's what you had. This was 300 images. But we're also getting the classes, we're also getting the score, the average score for it, and then how many objects are in the actual image in this case. So that's what we call the examine dashboard. Yes?
So really quick, if you go back to the grain image.
Sure.
I mean, is this, are you doing something other than basically a Hoff transformation? You're doing like actual just semantic labeling, or are you doing scale separation somehow?
No, this is YOLO V8 segmentation model.
Okay, so it's just a segmentation model. Okay.
Yep, it's an instant segmentation model.
Okay. And then are you able to like change your score, or is there a minimum that you have? And then you.
Yep.
Okay.
So you can have the default's 20% confidence score, and you can filter, but you can change that on the fly and filter out however much you want from there. So the 20 is the default, and we say, okay, that's good enough. But we can see that for these, we're around 92 average overall. But we can also see for on a per, this screen's a little small, but we have it per item too, per instance, right, has the score.
So this model is already trained by you guys, right? The
Yes.
YOLO V5 model. So how do
YOLO
you
V8.
handle the robustness? So how can you say that your model is robust against out of distribution samples?
I'm sorry, I
Is
couldn't.
your model robust against out of distribution samples?
Yeah, so does this work with, the question is does this work, how robust is the model?
Yeah, and also like, can we do transfer learning sort of.
Yep, that's exactly kind of what we did with it. So we started off with just a regular pre-trained ED class model, and then we just said, just train it on these 30 years worth of data that they already had, that they trained their UNet model off of. So the question was, for us was, okay, you have this UNet model that does just mass segmentation, doesn't do instant segmentation. YOLO V8 is very fast compared to the UNet model. So because we were like, hey, what if you could do this on the microscope? And they were like, we never even thought of that because we've never been able to get to that point. So we ended up going in and just taking their training set, converting it into a training set that YOLO V8 would on there. They are currently, as we speak, looking to see how robust is the model then for them. They're comparing it against the UNet for that. And then also they had their hand annotations that they had as well. So the question of is the model good or not will be answered in just a moment when we come back from the demo. Any other questions? Okay. So we call this the examine dashboard because this is for examining data after it's been allocated. But what we have over here on the left-hand side is all the different weights and models that we support. We currently support basically anything from the Ultralytics library, which is YOLO V5, YOLO V8, YOLO V9, YOLO V10, which was just released. SAM, which is a segment anything model. We were able to put that one in there. Based off of our architecture and what we came up with, that was released on April 13th of last year. And on April 29th, we were able to have that running on our system, entered into it. I only know those dates because my birthday is April 29th. So I was pleasantly surprised with a happy one for that. But we have a variety of models. We can organize this. If we don't see a model we like, we can upload one. And so this is all through, again, the web browser because it's running off of the web server that's on this device here. And we can go ahead and start a model. And so what this does is it's spinning up a, what we call an actor. We'll get into that in a little bit. But that's a model actor. It's loading it onto the GPU. It's going to start it up. We're idle, so we're ready to go. We can start doing an acquisition. This is a YOLO V8 Coco training set. This would be the default one that they provide. It's a medium size and it does segmentation. So we go ahead and start that. I'm going to do one thing here. And this. Start here. And so we start. Oh, did not like me moving that up. So it's in progress, and in just a moment, it should fire up, and we'll see on the right-hand side. Again, demos, who knows? Oh, because this fell out. Ah, that's why. Which is a bummer because
We just missed the good part.
yeah, because we are doing sort of running our model and running it on the everything in there. Right? So we're doing the instant segmentation. So you can imagine, I don't have an electron microscope here with me, so I can't show this in real time. So we use this as a demo anyways, but you're getting your scores are in there. I know it's a little fuzzy in there, but we're also picking up the cup. We can also...
Our scores?
Yes, how confident you are, how confident the model is that you are.
Oh, okay.
A person. So it's detecting 5 people, it's getting some backpacks, some chairs. We're getting our contrast histograms, we're getting all that information. But we also know that microscopists love to play and they like to change things. So we've added in a bunch of processing, just sort of what we'll call the classic model instant type. So this was the Kali normalization process that you can use. You can change your contrast, you can change all that. There's the confidence score you can also change. So if the lower confidence things you wanted to be able to change, you could go and you could change all the different parameters for that model. And the biggest thing for the microscopists has been, I didn't have to write a single line of Python code. So they're very happy about that and very excited by that. But we can go back now to the presentation, unless there's something else people would like to see.
Is this running in the cloud or is this actually just using
This device right there.
It's all, okay.
Yep.
And
It's
what-
just the interaction is through a browser.
Yes,
Okay.
everything is through the browser because we're using a web stack for that, and we didn't want to have the microscopists install anything on their microscope computer or even on their laptops because then they have to go to IT, and IT would be like, is it approved? We need to get approved, but the browser's already approved. So we can get around all of that and still use the web browser. So we have this thing running, so I just want to do one thing real quick. because we are here and I don't always get to play with this, so it is of course, here's the Cylon, so we can go ahead and do that, right? So, back to the presentation. Hopefully it doesn't take me to the beginning. Oh, good. So, that was a very, very quick and brief walkthrough for that, but what we'll come to is come back to the presentation. This is a video of, is it gonna work? I don't know why it's not going. Oh, there we go. So this is a sped up video of an in situ TEM experiment at MyBall, and we're looking at, now we're back to black dots, so not people, but black dots now. But this is also the kind of software that they're dealing with. This is considered modern software for them, where we see that we're getting all of our red dots at the microscope while we're running. It was one of these devices. It was sitting off camera over on that side, but it was one of the prototypes during this process. And what they're looking at, and what they want to know is this one, it's a little fuzzy there, but is that increasing? Is it decreasing? Basically, am I getting more defects or less defects as the radiation is impinging onto the sample at that point? Right, so they're getting that answer. That's the discovery. That's their interpretation. That's what they're trying to figure out. Is it a good material or bad material? That took them 2 years before. They're getting that now at the microscope. So let's see. So what we've learned and what we've done so far is that we know and we've seen that we can do pretty much anything that takes an image. We are no longer bounded by just black dots on gray backgrounds. We can do white dots on black backgrounds. We can do grain boundaries, and we can also do people and everyday objects, most importantly cats, right? So we can do it once, we can do one model. This presentation is about scale. So now, how many times can we do it? And the question that always comes up though is from all of these different applications, what model should I use? Right? So I've got all these different models being published all the different time. They're all for these different, it might be by material, it might be by camera that's being used, by detector. It could be by any, if you're in the bio side, it could be by cell line. There's all these different things. We're always asked, which model should I use? And our answer is, just run all of them. Just run them all. And let's see what happens, right? Let's scale up and let's see how many models can we run. But there are some problems with that, of course, that we have learned. Right? And this is one thing, which is your performance of your model is also hardware dependent, and it depends on the type of models you can run. It also depends on what kind of hardware you're using. Right? So this one here is the Jetson EGX Orin line. We actually originally started all the way back, if you remember, 45 minutes ago from when I started the conversation. That software that I wrote and showed him was actually running on a Nano. We were using the Xavier NXs for a while, but we and the EGXs, but those are no longer available. The only ones that are available are your AGX Orins. So if you hear me say the word Orin, that's where it comes from. So there's a lot of things that go into this when it comes to edge computing and why do we use edge computing and everything else. Because one of the things that a lot of institutions have possibly their own cluster, their own GPU, their HPC, so on and so forth. But there again, you're getting into that situation of, well, how do I get that resource to the microscope? Right? Even if it was on campus and on site, you're still going to run into bandwidth issues, you're still going to run into throughput problems, you're still going to run into IT restrictions when it comes to that. And what we have found though is that edge, or what we're calling near edge, like workstations that you could put in there, or rack mounted servers, are powerful enough and have enough to run the different kinds of models you want, and we can go from there. So the question then is, well, okay, some models do require more compute than others, so can we do ad hoc heterogeneous models, or clusters? So if you were fortunate enough to be an institution that had one of these on every single microscope, could we use them collectively together to create an ad hoc cluster and have, because like, oh, this microscope was down, but can I still use the GPU that was there to run my models when I'm over on this microscope instead, right? And can then the one that's connected to this microscope, maybe it's an Orin, but this one up here is a workstation, right? Can we still pool all those resources so that we're maximizing the hardware? Well, the answer is yes, you can, But in our context and for our software, can we do that? And the question isn't really it's not binary, it's how many can we do? And how many different kinds of hardware can we put into the system? So we started off with a very naive approach to this. And this was work we were doing with Idaho National Lab on this. And we come back to our platform that we came up with, and we said, well, let's just add a bunch of devices to the system. Let's just connect it all up. We have 5 of them connected to the microscope at Idaho National Lab. It turns out we can only get to 3. We ran into this 3 model limit problem in our system. And it was, which was fine. I would have preferred more, but you'll see what's happening here is that this one will stop reporting values here because what's happening is you're getting congestion in the system, and as we're running all 3 models, because remember we said, hey, just run all the models, right? Well, we tried to run all the models and it died. And so it's like, well, wait, how did that happen? Why'd that happen? Why can't we do any more models than 3? And what's that limit coming from? And ultimately that was because of our naive initial approach, right? We start off with the initial approach, and the issue was that we had 3 separate acquisitions going on, and you're sending that one image from that one system 3 times across the system, and you're running that all through there, all on your gigabit switch, and even that wasn't enough. And that's because we're doing also the full screen image of the monitor. And so it was also resolution dependent on your monitor. They'd upgraded their monitors because microscopists love high definition video. So we were trying to shove that across the network 3 times over, right? We're basically essentially streaming Netflix 3 times across this on this poor little device over here, right? So we had to come up with a better way to do that. In retrospect, probably should have known that that was gonna happen when we started, but yes.
How big is the model on the GPU memory? How does it fit inside that? Like how large is it? Because I would imagine it would grow or
Yes.
be considerably large. I don't know how much memory the GPU and the Raspberry Pi holds.
So the orange, one of the nice things about the orange and one is
Oh, it's the orange Pi.
No, it's an Orin. It's a Jetson
Oh, I'm sorry.
Orin from NVIDIA. One of the reasons we do like those is they have 32 gigs of memory or 64 variant now that they have available. And so we are able to use that. Now that's shared with the CPU, but we're still able to get that. A gaming 4090 GPU that you could use for training only gets to 24 gigs. So that's, you have to kind of keep that in context. But the models, it all depends on what you're running. A YOLO V8 model, it has essentially 5 different sizes. A Nano, which is like usually only like 5 megabytes, all the way up to an extra large, which can be about 150 megabytes. The SAM is an example. The B size is roughly about 350 megabytes, but the larger ones are up to a gig. And we actually can't really run the gig ones on one of the Orins. We could run it on a
It's called Orin.
Orin, yeah, O-R-I-N, Orin, yeah. So it all depends, because one of the things you can also do in, it's not in this presentation, what we have done is fractionalize the GPU, so you could use one GPU and run multiple models, assuming you have that size on there. And in the demo, I didn't show it, but there's a pie chart that shows you how much of the GPU you're using, and that can also be used to say, okay, what size model to run. Yeah. But that was one of the experiments that we were trying to do, is let's run all 5 sizes and see which one gives us the best results. Right? Because the question is the trade-off, accuracy versus latency versus what can you actually run on the systems in that time. So this is roughly an overview of the software stack that we use. A variety of technologies. It's all on the web stack that's running on that device right there. One we're going to highlight here is Ray, which is the toolkit. It's the Actor framework. That's why you heard me call it Actor. It's in Python. It's what was used to train the ChatGPT model. And it allows us to do the cluster management and everything else. I mentioned Docker earlier. That's where we're putting everything on there. We use a system called Balena, which is an IoT fleet management system that has a specialized Linux operating system that is basically just enough to get you onto a network and then run Docker containers on top of it. So we are able then to push all of our dependencies and changes onto our devices remotely through Docker containers, which helped out with the dependency management and so on and so forth from there. And then at the very top, we're using a web browser and Grafana for the dashboarding and interfaces. But going back to that 3 model limit, what we said is, okay, what we should be doing is instead of having 3 separate or n number of separate acquisitions, we should have one acquisition governed by a head node, and that dispatches or broadcasts that image to the rest of the nodes. So that's where we have then Ray, and Ray has this sort of architecture where you have a head node, you have the worker processes, there's a scheduler, and then there's this auto scaler that will automatically dispense the jobs to the different worker nodes, and each then node or device in our ad hoc cluster becomes a worker node, and only one of them has to be the head node. And what we'll do is the head node will acquire that one image and then dispense to the various worker nodes. And so our architecture then looks like this. We have our head node, as I was mentioning, that Ray is controlling it. The models are loaded onto each one of the separate horns or onto one of the workstation GPUs or a blade server or so on and so forth. And so then we have a single acquisition to multiple models. So, does that work? And let's see. The answer, of course, is yes, because I wouldn't be presenting if it didn't. But we can see we're running now 5 models simultaneously, 1 acquisition. We can do bounding boxes, we can do segmentations. We have good models, we have bad models. But the question is, which model do I run? Run all of them, right? And we can do this multiple times over and over. I don't know, that slide was twice. So what we found then is we were able to run the 5 models. These are the 5 orange that are connected to the in situ TEM at Idaho National Labs. And we ran all of those and we saw that, hey, our best model for this particular sample and this particular run was this YOLO V5. So not only are we heterogeneous in the hardware that we can run on, but we're heterogeneous on the types of models that we can run. We can run, all of these were YOLOs except for the one up here, which was the massacre scene in Wisconsin one. We are in the process though, of course, of moving that over to a new system. So this is just kind of a recap of what I just said though, which is, yes, we are able to do that. We can also do this using what we're calling a near edge workstation, which is a Lambda Labs quad system that was 4 A5000s that were sitting in the lab. They just happened to have that laying around and they were looking for something to do with it. And we said, hey, let's put our system on there. Because I guess one thing that you can use with program money for is to buy GPUs with, right? So they had that laying around, we applied that, and look, it works. We get the same count. They were happy that they were able to get the same data regardless of if you're using one of the edge computing devices versus a near edge. There was some concern there that there would be different responses. Yes?
How are you evaluating or validating the performance here? Are you basically looking at, are you segmenting are you partitioning the training data and then the validation and so on and so forth,
Yes.
testing set, et cetera, and then you're just using like 30 years or whatever of this back catalog to,
Yeah, so the training set is split into a 70-20-10 split. So 70% for training, 20% for validation, and 10% for testing, which is kind of a best practices or a good start situation when it comes to training the YOLO models. Then we throw that onto the device and then we run a whole separate set, which would be like the videos or an actual experiment, and they decide, is that going to work or not for what they're getting. So that there's a graduate student at University of Michigan whose thesis work is basically like, how good are the models and what are they seeing and how does that all work? And to answer that question mostly, because there has been pushback, of course, of like, how is that model, as good as a trained microscopist?
I guess like the other issue, of course, is just the issue of novelty, for lack of a better term, because you unless you are entrusting a graduate student to create your training set,
Right.
You're not going to necessarily have the scalability across multiple models for that training.
Yes, yeah.
So I guess, how do you solve that?
You have to annotate more models and annotate more images. You can also use, for example, the segment anything model as a starting point to get your segmentation. Maybe you use traditional computer vision to at least start your training so you can get some of that done automatically. The other thing that the University of Michigan and Kevin's group is working on is creating synthetic data so that that can be fed into the system and they would know where it's at. On a bioimaging standpoint, you could tag and fluoresce. You could use that to know to automatically annotate your images that way, and we know that NIST has been doing that for a while now too. So you basically use another sort of, we'll call it dimension to look at that, or another analytical technique. Another one that they use in this case is eels, and they'll have that on the microscope, and they can use that to identify and say, oh, this region is what it's looking at. Grain boundary is important because the eels data looks a lot like the grain boundaries, and we say, hey, does that work? It kind of does work for that too. It's pretty interesting. So I mentioned Idaho National Lab, so I'll mention them again. But the concept was, okay, we broke through the 3-model barrier with this system. We were able to run this on multiple types of GPUs and multiple systems, but can we do this in a distributed manner across their materials and fuels complex out in Idaho Falls? And so the idea was that they have this, they have their irradiated materials characterization laboratory that has a variety of different kinds of material characterization. This is also where the irradiated samples locate. So can we have basically a bunch of these spread throughout that part, but then also call back to our blade server, which is about a half a mile to a mile away over a 40 gigabit network, and have 6 GPUs onto that. And then also can we shoot up that code up to a cloud-based system if we wanted to in the future? The answer is yes, we can do that. It did work. I had 11 GPUs. I ran 11 models at the in-situ GEM at Idaho National Lab a couple of months ago. So we were very happy about that, and that this basically helped us validate that at least our architecture when it came to the ad hoc heterogeneous clusters was correct and was on the right path to be able to run n number of models, where n is limited by how much hardware you have available to you. So with that, we had a variety of people that we've been working with over the years, but we've been deployed at, again, Argonne National Lab, we were out at Idaho National Lab, we were at Sandia National Lab, University of Michigan, and University of Tennessee, and then we were just recently out at Los Alamos National Laboratory as well. And so we were able to, this has been ongoing now for a better part of 3 years on that, and the team members that have been involved in this, I'd like to, Gabriella is the one that is working on the bias and is it a good model and the synthetic data part of that. Kip Wheeler is working on the decision tree for figuring out if the model is good or not. I like to just make a special call out to Mikhail at Volkov Labs. He does all the front end development work for us and has put all together, so the dashboard and everything you saw earlier was from him, so that was great. Fantastic. And then we had a variety of collaborators over the years.
Excuse me. When you said bias, what sort of bias is being, are you guys treating in the machine learning models?
Yeah, so the bias is coming from the user that's annotating the training set images originally. Right? Are they seeing the same thing? Right? We saw that there's the wide error bars between the users. So what bias is there, and then how does that propagate into the models?
Okay.
And then how much bias can we tolerate in order to get to the answer that we ultimately want, which is this a good material or a bad material, based off of the metrics that we're looking
How
at.
do you mitigate the bias in this?
That's something they're working on. So you try a variety of different models, and you also have as much variety of annotators as possible.
Right, but I know we employ pruning techniques for removing bias, because bias is being hidden in the neuron level of the machine learning models. So certain neurons in the model will be susceptible to bias more than other neurons.
Yes.
So that's why we have more overparameterized networks for more robust generalized models. But how do you, okay.
That's a much broader question than, and that's on the development side of that ML sort of workflow, whereas this work at least is just focusing on the deployment, right? And deployment from the perspective of the microscopist who's like, I just don't want to have to draw boxes from black dots, right? So that's where the research is coming in, and that's where University of Michigan is doing that, and I will just have to direct you to Kevin to answer that question.
Thank you.
So with that, thank you all. And all of this work was funded through SBIRs with the Department of Energy, so thank you to the Department of Energy for all of that. And then here again is all of our different workers for that. And we have a little bit of time, or if you stick around, you can do this now too. We have it set up. If you look on your laptop, there's the Theiascope 5G or the Theiascope Wi-Fi connection. Here's the password. You can log in and give it a shot if you'd like.
Okay, well, let's thank our speakers.